Can you hear me? Oh, oops. Yeah. Okay. Good. All right. Thank you. Let me start by thanking this great university, really, and the amazing institute that you have founded here, and for the stimulating conference that you've organized, and really, most importantly, also for taking a principled stand against authoritarianism. I think we all owe you a huge favor. You're suffering really on our behalf, on all of our behalf, and may you flourish for the next 400 years. Okay, so thank you. Yeah. After Kunal's talk, I really should probably just walk away after the slide. No, just kidding. All right, so today I'll tell you some ongoing work where we try to deal with causality in a very small way. And this is focused on trying to figure out how biological learning works in the brain. This has been really a topic in the last couple of talks and others earlier. And several of the speakers have actually contributed to this topic over the last few years in work that they didn't talk about, so quite apropos. This work has been spearheaded by Kevon Day, Martin Rosa, Christina Wang on the experimental side, and Kyle Aitken, Matthew Bull on the computational side, who has also help from Matt Gallup at UW and Stefan Mihalas at the Allen Institute. We've, over the years, had a really great kind of discussion group going with Google DeepMind, in particular in the early days, Tim Lilliecrop, Peter Humphreys, Matt Botvinnik, later Claudia Klopath, Zeb Nelson, Kim Stachenfeld, Maria Epstein. They all contributed intellectually to this work and to kind of governance that I don't have time to tell you. So, imagine you want to learn how to play the piano, tune on the piano, right? This is really about making complex sensory motor associations between, you want to make associations between sensory input, for example, notes on the page of the paper or the sounds from a piano, and link that to the motor output, the next placement of the fingers at the right time and right place. So as you probably know, abundant evidence, and so we already discussed, suggests that learning is associated with changes in synapses. And there's tons of synapses, about 100 million synapses per cubic millimeter of gray matter, cortex or something like that. And synapses are embedded within multi-level neural structures as they are in artificial neural networks. Like, for example, imagine motor learning, which you've already heard often happens in superficial layers of the cortex. It might be 4 or 5 synapses away from motor output and 4 or 5 synapses away from input. Okay, so moreover, any synapse in the motor cortex, tweaking any synapse has only a tiny impact on the output of the nervous system onto behavior. Okay, so how does the synapse in the middle of the brain then know how to change to advance a behavioral goal? And this is really what I'd like to talk about, and is also referred to as the synaptic credit assignment. Now, in artificial neural networks, of course, powerful, and I really don't need to belabor this for this crowd, powerful methods exist to solve the synaptic credit assignment problem. Learning algorithms really generally improve network performance to achieve a particular goal through a loss function, right? And backprop of error is such a learning algorithms, right? It computes how changing each synapse in the network really changes the network's behavior, and changes of synapses in one part of the network is really aware of synaptic changes throughout the network. So as a consequence, the backpropagation error is a powerful vector feedback dominated algorithms that moves up the gradient quickly, gets you to the goal quickly, but is thought to be biologically implausible. Now, other mechanisms have been proposed that are biologically more implausible but less efficient. We've already heard about Hebbian plasticity. What I mean by Hebbian plasticity is simply the kind of plasticity that is strictly local between synapses, right, related to correlations of pre- and post-synaptic activity, which then leads to a synaptic change. There's a large class of mechanisms even in there. But in general, Hebbian plasticity defined in this way by itself does not know about behavioral outcomes and thus can't improve network performance to achieve a behavioral goal by itself. Now, three-factor learning is again a class of mechanisms that is Hebbian in nature with a global scalar feedback signal. So it's a learning rule that often is thought of to operate by trial and error. Neural activity, right, perhaps driven by noise or some random input, might modulate a behavioral output. The correlation in activity, the Hebbian process, produces what is called an eligibility trace. And the third factor, which could be, for example, neuromodulation or something else, then depending on the behavioral goal, captures the eligibility trail, potentiates or depresses a synapse, right? And the third factor, of course, has to report if the output is getting better or worse. So the learning rule keeps synaptic changes that are consistent with improvement and discards other synaptic changes. So three-factor learnings can be slow, but in many situations will eventually train the network and allow the system to go up the hum to the behavioral goal. So its learning resembles kind of a gated random walk. As you know, the brain, of course, generates diverse signals, kind of scalar-like signals, right, that monitor behavior performance, positive reinforcement for the correct phrase of movement, or for the correct phrase of music, negative reinforcement in the case of dissonance. Midbrain dopamine neurons are an example of such a signal, but there are other ways for, as I've already mentioned, for performance signals to get into the brain. Now, in addition, and I won't talk too much about that, you've heard a little bit about that already, there are biologically plausible vector feedback learning that have been proposed, often algorithms that invoke dendritic mechanism, actually by Zhao, Sacramento, for example, and others, error or feedback modulated Hebbian learning, and so on and so forth, that eventually can essentially approach kind of backdrop-like performance. These feedback signals that are required, okay, now for these kind of more sophisticated learning algorithms need to identify neurons that had played particular roles in driving a behavior and require feedback signals that really look a bit like the error signals that you see in predictive coding frameworks, right? They're present in the brain. We don't know yet how they feed and might modulate learning deep in the brain. In addition, this kind of learning requires complex and specific feedback circuits. So overall, there's been a lot of theory, lots of learning rules have been proposed that shape behavior-related plasticity, and we'd like to figure out how this works at the level of biological hardware, mechanism, really. So why is this hard? One reason is that representations of behavior-related information is widely distributed across many brain regions. And memory traces could be widely distributed and sparse in any brain region. So I just want to point out a study from Ran Darshan that is cited there, who showed, for example, that tuning in recurrent neural networks, that if you tune just a small fraction of synapses on individual neurons, small proportion of synapses, you can change behavior-related activity in profound ways, and these changes propagate through unmodified synaptic pathways to change network activity profoundly, even though only a tiny proportion of synapses and a tiny fraction of neurons have been changed. So to overcome this kind of needle in the haystack problem, we developed a simple brain-machine interface task for mice, where neural activity in a bunch of neurons that we choose as experimenters control an external effector. This can be manipulandum. In our case, it's very simply the proximity to a reward port. And this kind of BCI gives us access, of course, to neurons that are causally involved in controlling the task. These are the yellow triangles there in that cartoon. And the experiment, in addition, of course, imposes the mapping from neural activity movement of the manipulandum. Here, in our case, the conditioned neuron, the BCI neurons, simply drive the distance or the propagation of the reward port towards the mouth of the animal. And the experimenter also controls feedback about the approach of the reward port, sensory feedback, and reward feedback. The job of the brain is really to learn how to modulate the chosen BCI neuron, or BCI neurons, to control the effector. And this paradigm really allows us to link learning, network plasticity, and the factors that drive plasticity in the brain. So well, in the next 15, 20 minutes, I'd like to give you 3 short related vignettes. I'll first describe learning and dynamics in a simple version, the simplest version of this motor cortex BCI tasks. And what we find is that changes in activity can be remarkably specific and sparse, providing, I think, challenges for learning routes in the brain. We then compare changes in neural activity and connectivity in the brain to artificial networks trained with specific learning rules. And we think that these comparisons suggest that the learning that we see in the motor cortex and BCI learning might actually be supported by a 3-factor learning task. We then combine the learning in addition with a causal mapping of circuit structure to identify the changes in the cortical networks that happen, again, providing additional evidence for 3-factor learning in this task. And so Conrad's point is this is probably not, what is it called, causally sparse. So we will see but we're trying to push this kind of hypothesis as far as we can. Okay, let's start with the brain-computer interface. This is actually based on our paradigm. The paradigm that I'd like to propose here, I'd like to show, is based on a task that Epfetz developed some 50 years ago. Okay, it's very simple. The animal has to modulate a particular neuronal activity. It's a single neuron that we pick out of a hat. It simply has to be a neuron that's active, but that does not show any task-related activity. The animal, after trial start, has to modulate the activity up. That drives the approach of a reward port. Then it has to modulate the activity down to receive a small water reward. Okay, it's very simple. All the while, the proximity of the reward port is accessible to the animals through somatosensation. It's important for learning this task quickly. We use imaging to track activity. It's sort of a low-pass filtered version of spike rate, if you'd like. This is because we'd like to identify not only the conditioned neuron that we track by imaging, but we want to see what neurons do in the vicinity in the motor cortex network. And imaging also allows us, because we know what the locations of neurons, to manipulate neurons that are part of this circuit that involve the conditioned neuron. Okay, it's kind of important for this approach. So here are some behavioral trials. You have a couple of trials where the animal failed. Okay, you see the reward port comes not quite to the threshold where it's accessible to the animal. That you can see that's related to the small amplitude of activity. In the first couple of trials, then the activity kind of ramps up. The reward port moves more quickly, and the animal is successful in gathering reward. So let's look at one experiment here in action. Okay, so here, there's a lot happening in this visual field of view. There's this calcium imaging there. You see these neurons that flicker on and off. This corresponds to small bursts of action potentials. A lot happens in the conditioned neuron that is circled here by the red circle, and other neurons in the vicinity, okay? But only the conditioned neuron really matters for the movement of the reward port, okay? So you can see a few successful trials. Green circles refer to mark the trial start. After trial start, the activity goes up, propels the reward port forward to get the animal the reward it seeks. Okay. So in sessions with successful learning, activity of the conditioned neuron rises quickly, right, over a couple of seconds, usually to drive the reward port towards the mouse. We refer to the modulation of the activity of the conditioned neuron as tuning. Okay, the modulation of activity after trial start as tuning. And you can see that in this session there on the right, that it took a few trials, right, for the conditioned neuron to kick in. Okay, you see sort of trials in the vertical dimension on the right, trial time within a trial on the horizontal dimension. Few first trials, the animal doesn't know what's going on. It understands the task, right? It sort of figures out this neuron is the one. The neuron starts firing, then leading to successful rewards. And after a couple dozens of trials, the animal is sated, stops working, and we go on to the next behavior session. So on average, mice learn this task quickly. They become experts after a few sessions. And once they're experts, it takes only a couple of tens of trials, often 10 trials for the animals to learn substantially. They learn this reliably and quickly, and leading to an increase in the reward rate, but also the rate of reward in time, because the higher the activity, the shorter the trials are. There are many ways, of course, to solve this task. The animal could just simply modulate the activity of many neurons in the motor cortex. But when you look at the changes in activity, this is now a movie that I'm about to show you, where you just show one image per trial. And the image that you'll see is the change in activity compared to baseline activity. Okay, and like in the experiment that I showed you earlier, the conditioned neuron sort of slowly comes up. It's here under the magenta neuron, but very few other neurons really change their activity pattern. Right, it somehow very quickly figures out how to identify the conditioned neuron, with just a couple of other neurons coming along for the right there in the lower kind of lower center of the field of view. Okay, so incredibly sparse learning underlying this kind of BCI learning. But somehow the mouse brain identifies the one neuron that we happen to pick with a couple of other neurons that come along for the right. So these learning-related changes are sparse. There's, of course, nothing in the task that requires the neural activity changes to be sparse. There's a powerful synaptic learning rule that underlies these rapid and sparse changes. Let's look at this sparseness in a couple of other ways. Here is kind of the manifold on day 1 and day 2, the activity manifold on day 1 and 2. The magenta arrow indicates the conditioned neuron. You can see it's a blue streak. It's a bit hard to see on day 1. And then the activity on day 2 after BCI learning. So it's kind of baseline condition on day 1, day 2 after learning. That blue streak has turned into a high activity streak. And you can see in that scatter plot that corresponds to this one neuron that changed its activity quite a bit across days, right, from day 1, the baseline day, to day 2 after BCI learning. So, in most experiments, the BCI neuron is in the top couple of percentiles of neurons that we image. Of several hundred neurons, it's sort of in the couple, 3 or 4 neurons that we see. So sparse learning balances flexibility and stability, fast learning in a small proportion of neurons, but overall the population activity remains stable. Okay, so how might this work? So what kind of learning rules can produce the sparse and rapid learning? We start by comparing changes in neural activity and connectivity in BCI to artificial networks trained with specific learning rules. And we implement these learning rules here in standard RNNs. Here we start with a 3-factor learning rule. And the learning rule is shown right here. The change in synaptic weight is related to some kind of representation of reward compared to expected reward, some kind of time average with change in reward, and then eligibility trace that corresponds to postsynaptic and presynaptic component. The postsynaptic component has this 5-factor that just makes sure that the eligibility trace only accumulates if the activity of the postsynaptic, if there's activity in the postsynaptic neuron. So the third factor is really this reward prediction error kind of signal, which might be produced, for example, by a neuromodulator or some other relatively broad scalar signal in the brain. So in this kind of simulation, weight updates have no knowledge of what the conditioned neuron is. The animal has to figure out, or the brain, or the network in this case, has to figure out what the conditioned neuron is by trial and error, by exploration, this kind of a reward enforcement learning. So here's what we see in the recurrent neural network trained with a simple learning rule. In red is the activity of the conditioned neuron that we picked in kind of faint red, the average of other neurons. There's this specific change in activity in the conditioned neuron that leads to an increase in the hit rate and in the reward rate acquisition, right? Because also the trials are getting shorter. And the learning rates are sort of similar to what we see in the experiments here, comparing the blue and the black curve on the right. Importantly, the 3-factor learning also produces in the RNN network sparse activity changes. Okay, so this here on the right, the scatter plot showing the activity of the conditioned neuron on day 1 versus day 2, both pointed out by these errors, and the change in tuning by the of the conditioned neuron, which kind of rises out of this funnel of noise describing the fluctuating activity of the rest of the network. And both in the biology and in the model, the network response is sparse. The conditioned neuron is often the top changing neuron in both kinds of networks. Importantly, or in contrast, networks trained with some of the kind of gradient-based methods, these approximations to backprop, for example, feedback alignment, don't produce sparse changes in activity, because these systems don't, these kind of algorithms don't have any way to figure out, right, through trial and error, which one the conditioned neuron is. Now, so this provides, I think, some evidence for 3-factor learning. At least this means that 3-factor learning plausibly analyze the changes in activity and learning during BCI learning that we observe. But however, it turns out that this 3-factor learning would operate on the network that we observe, or on upstream neurons, so maybe on thalamocortical synapses and pinch, so upstream synapses that impinge on the network that we observe. And in fact, upstream learning with 3-factor learning or training the network itself both can produce similar learning rate and the similar kind of sparse activity patterns that we see. So to distinguish between the possibilities, we now measure learning-related changes in network connectivity using 2-photon optogenetic and imaging. Okay, so the way we do this, you've probably heard about these methods. We retain the ability to image neural activity by calcium imaging, but also express kind of a optogenetic switch in these neurons, which allows us to activate almost individual neurons, maybe 1.5 neurons or something like that on average, and then look at the responses, the postsynaptic responses to stimulating these neurons. And we refer to this as causal connectivity to distinguish this from kind of correlation-based measurements. So because we're looking at the postsynaptic response, right, on activity of other neurons evoked by spikes that we add through optogenetics to particular neurons. And I should say that the effect that we observe is not synaptic potentials, but a change in the spiking probability of downstream neurons. So that's what causal connectivity measures. Okay, so here, there on the right, you see the excitation of a target neuron and a few downstream neurons, a couple of neurons that are excited in a with an excitatory postsynaptic potential, one neuron with a inhibitory potential, likely through indirect GABAergic pathways. I should say all of the imaging here and excitation is done on excitatory neurons. So we can do this very rapidly and get a picture of the network connectivity. On the right is the kind of output you get from these measurements, where each line here, each red line, corresponds to the strength of a connection to different neurons, and the blue circle indicates the amount of outflow from individual neurons, okay? And the kind of structure of this connectivity is consistent with what we know about the connectivity, the synaptic connectivity in cortex circuits. It's mainly for a relatively short range, on the order of a couple of 100 micrometers or less. So how do we now weave this into the BCI experiments? It's quite simple. We do BCI experiments every day, okay? And after each BCI experiment, we do a circuit mapping involving the same neural network. And so we get a kind of a representation of Wij on day 1 and day 2, and we can subtract these weight matrices. Remember, there's causal connectivity. It's not strictly synaptic strength. It's something that's related to synaptic strength. We measure the change in synaptic strength that is induced by BCI on day 2. And we get some really interesting patterns of connectivity changes. For example, neurons that are enhanced tend to connect to each other. Okay, so this already seemed to us like a strong evidence for local plasticity in the neural network, that we observe. It turns out it's actually that can also be produced, this kind of pattern of connectivity change, by long-range input, right? Because these are nonlinear units, right? And just by changing kind of where you are on the activation function, you essentially can see things that look like synaptic changes by virtue of this causal connectivity measurement. But additional evidence comes from comparing Wij, the strength of synaptic connections and correlations, okay? You know about noise correlations. Noise correlations measure the fluctuations of activity when you take out kind of task-related modulation. And it turns out that noise correlation predict causal connectivity in these networks. This has been shown by others before us. More importantly, for the purposes of the point I'm making, right, is that the change in causal connectivity produced by BCI is associated with a change in correlation. And this kind of relationship we have not been able to reproduce in networks that are trained with where really the input to that network goes synaptic plasticity, or where an upstream network is plastic. So I think this is kind of strong evidence for local plasticity in the observed neural network. We can look at to test specific predictions of three-factor learning. Okay, one prediction of three-factor learning is that correlations during the BCI learning should predict changes in connectivity. Give you a couple of examples of individual experiments. That's in fact what we find. Okay, so there is a change in connectivity for this kind of bint data and plotted against the noise correlation during BCI learning. You can see this positive relationship. Stronger correlations mean larger changes in activity. We refer to this as positive Hebbian index because it's kind of positive Hebbian, right? The correlation, more correlations lead to larger changes in weight. But it turns out that the Hebbian index, the slope of that curve varies from experiment to experiment. It's even sometimes negative, right? So here is an example where the change in activity actually decreases with the strength of connections. And this is exactly what you actually is exactly predicted by three-factor learning as follows, right? Remember that the form of the learning rule, there's a factor that's proportional to correlations, right, and monotonically related to correlations. There's this kind of reward prediction error term, right? Is the reward better or worse than I expected? And if the reward prediction is positive, the change in synaptic weight will be positive and will increase with increasing correlations. In contrast, when the reward prediction error is negative, it will decrease with the correlations. I recall that in our task, we can test this directly because sorry, I'm almost done here. Because the reward is actually controlled by the activity of the conditioned neuron. It's the activity of the conditioned neuron that drives the reward part towards the animal, and that predicts reward. Of course, the faster the reward part moves towards the animal, the higher the reward prediction, right? So we can kind of replace here, oops, sorry, I'll use this as a pointer. We can kind of put into first order the activity of the conditioned neuron in there for reward in that expression, and then it turns out that conditioned neuron activity at the start of learning really should predict the Hebbian index, and that's indeed what we find. If the conditioned neuron actually has a little bit of negative tuning at the start of the learning, the reward prediction is negative, and we have anti-Hebbian learning in the network. Learning is slower, and so on and so forth, and gives this kind of negative relationship between change in synaptic weight and noise correlation, and the converse if the conditioned neuron tuning is positive. And this holds across the population, both at the level of experiment and in the modeling data. So what I've shown you is that we have rapid and robust learning, and I think this is important because there is a lot of experiments we need to do to map circuits, but also to get at neurobiological mechanisms ultimately of learning. We want to do this efficiently. Lots of stuff to do. Changes in neural activity during BCI learning, the local synaptic network changes, right? And because the local synaptic network changes, we decide where to look in the brain. Of course, the mouse doesn't know where we look. It doesn't know where we look. So likely these changes in activity and changes in actually plasticity are widespread, okay? So synaptic changes are rapid and widespread. There's sort of interesting questions, right, about how do we keep synapses stable, synaptic network stable in the face of this change. And the synaptic changes that we see are consistent with a three-factor learning rule. Okay? And we think there's a really powerful system really to explore synaptic learning deep in neural networks. I just wanted to highlight two kind of directions where we're taking this now. Is one, of course, is to change the task at hand to make the learning harder in ways that it can't be learned with three-factor learning rules, right? So that the kind of the gradient learning, the kind of directed kind of updating of synapses might have to happen, and we're kind of prototyping a bunch of kind of learning paradigms. So there will be at least a fourth factor in that learning rule, a kind of a factor that might direct learning in a kind of neuron target specific manner, right? Some kind of vector feedback. And then the other way we're trying to put meat on this mechanistically is to go in and look at the synaptic mechanisms that underlie this kind of learning and prepare particular state variables that we know operate at the level of synapses, again, to link these kinds of mechanisms to now cellular and molecular kind of factors. Thanks. Be happy to take questions. Yeah.
talk. So the motor region's really obvious to try, but have you thought of trying a more indirectly connected region? Because then I can imagine the learning is slower and maybe causes a larger population change if it can do the task. And that might be interesting.
We have not. There's a literature in BMI to look at conditioning and efficiency of brain-machine interfaces in across brain regions. And yes, sort of motor cortex regions tend to be a little better, but not hugely better. But we haven't done this systematically, right? So sensory cortex would be fun to try.
Great talk and really kind of stunning work. But to what extent do you think the particular structure of the learning rule that you're finding is particular to this kind of artificial BCI task? And more broadly, if you could recklessly speculate, do you think the brain uses one learning rule or like a couple or many?
Yeah, I think so let me just be clear about this. I'm actually serious with respect to the point to Conrad said. This is sort of, it's causally not sparse, where there could be gradient mechanisms going on here that we just don't know how to look for, right? So we may still miss some things. Even all of the predictions that we've come up with, right, for how three-factor learning rules should look like, should reflect in changes in the structure of correlations and connectivity. There could be things that we're missing. So I think that's one reason actually that going to kind of cellular levels is important, right? So for example, we have a pretty good idea about how Hebbian kind of plasticity should work, at least between pyramidal cells, right? And so if we kind of, in some ways, manipulate these things to establish causality between these cellular processes and learning, we kind of make a step forward. So, but more broadly, there's no question that there will be many mechanisms, and we know really from, for example, fly work, right, that there are lots of stuff that's going on in certain areas of the brain that are really two-factor, that only depend on presynaptic modulation. Just based on what we've seen in biology over the last 20, 30 years, I think one would be foolish to dismiss those kinds of learning rules. On the other hand, I think that some of the rapid learning we see even in mice under some conditions are not going to be compatible, of course, with three-factor trial and error learning. So some kind of approximation to gradient learning will also have to exist. And so I think for us, really, the race is on is how do we how do we kind of build the right paradigm to explore these kind of, look for these gradient mechanisms, and how do we actually, what kind of measurements do we do? I think we'll have to look at the synaptic inputs to individual neurons during learning to detect kind of more directed ways of modulating learning at the level of individual cells. Finally, about motor cortex, about this particular learning versus other learning, this is something we haven't gotten around to doing, but one could easily try standard kind of motor learning tasks and look at these kinds of, now we know sort of what to look for these kinds of hints of three-factor learning in more classic motor learning, actually the type of stuff that Mackenzie talked about. Yeah.
Thank you so much. Quick question, this is a modeling question. In the case of the local synaptic connections, I don't know, do you measure the number of synaptic jumps that is common? In other words, are we talking about signals propagating 3, 4, 5, 6?
That's a good question. I should have mentioned this. This is largely one synapse. Okay, so just by kind of how the structure maps onto the known connectivity profile in cortex, and we're stimulating the network very sparsely, and so adding a couple of spikes to individual neurons, right, is not going to cause a huge amount of propagation through many paths through the network. So I think it's largely directly coupled neurons. But I've shown you also examples of a GABAergic response. So there's some indirect stuff as well. So yeah, but mostly excitatory one jump. Sorry, you.
So you mentioned that it's a reinforcement learning type algorithm. Where's the noise being added in that? I didn't.
But there's some noise, there's some kind of variability that comes into the network.
I see. And it learns only from the noise component, or it learns from the whole thing?
It learns from the whole thing. Of course, there's sensory input, right? There's a trial start. There's information about the distance to the port, which is very important, of course, for learning. If you don't get sensory feedback, actually, FETs already figured this out 50 years ago. So yeah.
See, but here's my confusion. So if I have a neuron that has a positive influence on an action, then these rules basically become a version of gradient descent. But if the neuron has a negative influence, it becomes the opposite of gradient descent. So among a lot of people I talk with, like the simple three-factor rule is kind of like a null model. It's kind of like it can't work fundamentally because the neuron doesn't know if it has a positive or negative influence. How come that works in your simulations?
I'm not sure. I mean, this learning rule, reinforce is in some sense, right, the OR three-factor learning rule. It just out of the box doesn't work in neural networks. So this learning rule is very simple. There's a loss function, that's reward prediction, and it kind of maximizes. It tries to reward large fluctuations, positive fluctuations in activity, and then you expand it and just keep the local terms, and that's the three-factor learning rule. Yeah.
So you're then in the noise-dominated regime. No, like if it's mostly noise, then it's just perfectly gradient descent what you implemented. If noise is pretty low, it does gradient descent or anti-gradient descent.
Okay. Yeah. Maybe. Yeah. Yeah. No. One understands this a little bit better. Yeah. Yeah.
So how can you make sure that the connectivity change is actually responsible for increasing the firing of that neuron? So another scenario is other mechanisms are actually causing the neuron to fire, but because of the correlated firing and maybe prediction error, because of that, these are present, it happened to increase the connectivity, right? So what is, maybe related to Conrad's argument, knowing what is actually causal to the change of that neuron.
Yeah, we don't know that. We don't know that for that neuron. We can't map enough connections. When you saw these plots, right, that show correlation, that's for all neurons, right? But the number of connections that we measure just for the conditioned neurons are very few. And in fact, there is even this slightly paradoxical effect that I didn't mention, is that the overall input onto the conditioned neuron actually decreases. We don't understand that yet. So what causally makes the neuron fire, the conditioned neuron fire, I don't know. I can just say that many of the changes in connectivity in the local network are sort of consistent, with kind of a three-factor learning rule. And of course, local contributions and long-range contributions that ultimately drive the condition. Yeah. Yeah.
Thanks, Karel. I have a question with following up that the prior question, which is basically, so let's say you're saying that this is very fast, this sort of like learning is very fast, and you're showing us a conditioned neuron that basically initiates a cascade of other set of neurons, very sparse, to become active and execute whatever it is. So maybe one way of looking at it is not to say that these neurons are learning to do something, but rather they're actually being pulled into an assembly, which is already has that knowledge. So that prior is already built in because, this rat or mouse lived before and has learned to execute a set of certain things. But what you're testing now is just you're testing and see which group is pulled into executing this function. So because you're not seeing the input into this set of things, and rather actually your measurement is the measurement of output of this set, then you will be blind basically to see that sort of learning emerging.
As I mentioned, there are things we don't see here, right? What we do know is that the local circuit involving the conditional changes, right, in a way that is predicted by three-factor learning rules. So there are synaptic changes. Number one, they're measured, right, and they relate to a kind of reward prediction that the animal sees, and to the structure of the correlations in the network. So it is really related to Nero's question, exactly what makes the conditioner on fire. We don't know in great detail. It may be local and long-range input, but it would be given, right, that most connectivity is local, right? It would be a miracle if sort of local circuit changes don't contribute. Yeah, the local circuit changes that we observe and measure. As I said, what is it called? Causally not sparse. So yeah, good. That's it. Thank you. Yeah.